Hi, everyone. I'm your host, Rob Culverhouse, Associate Professor of Medicine at WashU. Welcome to ICTS Precision Medicine at Venture Café. We meet on the third Thursday of the month at 4 p.m. The sessions last 50 minutes, typically to allow speakers for the next session to set up, but we could potentially run a little bit extra today if we need to. As always, our mission is to connect the innovative biomedical science generated by WashU, SLU, and UMSL with entrepreneurs and businesses in the St. Louis region. We hope these sessions will stimulate your thinking, and we encourage you to share your insights and ideas with the speakers after their presentations. I'll mute the audience at the beginning of the presentations, and afterwards, when you have a question, please just unmute yourself. Today's topic is brains and chips, and our speakers will discuss neuroscience insights uncovered from neural recordings and how these insights can be translated into biomedical science and into computer chips. Our speakers are Drs. Keith Hengen and Dr. Shantanu Chakrabartty. Keith is an assistant professor in the Department of Biology at WashU. His lab investigates the neural networks in the brain and the interaction between disease and disruption of these neural networks. And Dr. Chakrabartty is the Clifford W. Murphy Professor of Electrical and System Engineering at the McKelvey School of Engineering at WashU. His research explores unconventional analog computing techniques using silicon and hybrid substrates, and he's using these novel techniques to design self-powered computing devices with biomedical applications. And I think today he'll tell us something about what he's learned from these neural networks and how he can apply them into designing better or different kinds of computer processors. So anyway, first up is Dr. Hengen, who will discuss emerging tools and computational approaches to neurological diseases such as Alzheimer's. Take it away, Keith.
Rob, thank you very much. And I have no idea who's here because this is Zoom, but.
And just a second, I'm going to mute everyone, and if you'll just unmute yourself. Thanks.
So thank you for being here. Hopefully this goes better than my lab meeting. This morning I was talking and door. This is Keith. I'm in the Department of Biology, been at WashU for 3 years. And I guess that I'm broadly interested in how it is that the brain is the most sort of effective, efficient, powerful computing device that we know of in the universe. I mean, a human brain is 86 billion neurons that somehow self-assemble without very much instruction, and they give rise to everything, your loves and your hates and your hobbies and your vacations and your memories. And we do this day in and day out without failure. And I will never mistake my car keys for a grapefruit. And if I do, I would argue that that's not normal, that would end up being kind of the line between healthy and severe pathology. And so we just sort of expect these things to robustly work all the time, and yet we have very little insight into how that actually happens. So again, I apologize to the neuroscientists in the audience. I'm going to jump over a lot of specific details, but I think that many of the reasons that we don't know how this kind of emergent property of computation works is because we've been limited by tools, and we have a tendency to embrace the sort of philosophical pipeline of reductionism. And so the name kind of says it all, but reductionism is the tendency to try to explain some complex system in terms of sort of simpler, smaller internal components. So if you think about the experience of driving, at a reductionist approach and trying to explain that would be to say, break the car down into all of its constituent pieces, and then maybe you could go in and eliminate this belt or cut out that bolt or throw out this screw, and eventually you say, aha, I found it. The spark plug is the essence of the experience of driving, right? And that would kind of be the reductionist approach to understanding what's pictured on the left here. And reductionism is certainly very, very effective in some cases, and it's certainly very, very not effective in many, many other cases. And as I'm going to sort of present to you today, I think that reductionism is let me try to say this tactfully, a leading cause of failure in our ability to cure neurodegenerative diseases. So reductionism in the brain takes a similar form as to what I just showed you. It might have seemed a little ridiculous in the car example, but bear with me. So here, can you see my mouse if I move it? Is that visible to you? Somebody shake their head. Yes, okay, great. So this is a coronal section of a human brain. So if I were to take a chainsaw and cut my straight through my head this way, that's roughly the outline of my brain. And the idea here is that here you can see there's a healthy neuron. It is labeled normal. You'll have to take my word for that. But then in neurodegenerative disease, things look a little bit different. So specifically Alzheimer's disease, the neural tissue is the defining feature of Alzheimer's disease is the presence of two nasty molecules. So there's these amyloid plaques, these A beta plaques, and you see these kind of gummed up just junk between the neurons. And then within neurons, there's these neurofibrillary tangles of tau. And tau is observed in many different neurodegenerative diseases, but it's the A beta and tau together that are the definition of a brain with Alzheimer's. So we have spent a lot of time and money trying to pin Alzheimer's on these two molecules. And I'm not saying that they are not toxic and that they don't kill neurons. I mean, they almost certainly do. But I would like to sort of present an alternate case to you for a moment. So Alzheimer's as a business community is probably worth considering because many statistics, and I'm sure you know some of these, but AD, Alzheimer's, is the 6th leading cause of death in the United States. And I believe that of the top 10 causes of death that are diseases, it is the only one for which we do not have an effective intervention. I read earlier that in 2016, just the caregiver costs from AD amounted to $250 billion in America. In 2015, Alzheimer's is projected to cost us $1.1 trillion. And this kind of blew my mind. Since 2000, heart disease deaths have fallen 14%, but deaths related to Alzheimer's disease have increased 89%. Right, so this is certainly a costly and pertinent problem, even if we ignore the kind of emotional trauma of the fact that it's going to affect almost all of us, either personally or by 1 or 2 degrees of separation. So with that, as I mentioned, we've been very aggressively targeting tau and A beta, and it doesn't seem to work. We've spent decades and hundreds of billions of dollars building antibodies against these, trying to sort of vacuum them up and sponge them up and sort of stop their production. And just last year, there was a trial at Roche that a lot of people had kind of pinned their hopes on, and that failed. And I guess I would like to suggest that perhaps investing another 10 years and hundreds of billions of dollars in another pass at tau and A beta might not be the most effective use of our time and money, or at least the kind of hyper-reductionist approach to these molecules. And so the reason for that, there is some evidence that I'm not crazy. So I'm going to suggest this systems view of neurodegeneration. So the idea here is that in contrast to reductionism, there's an idea of emergence. So when you take a lot of small pieces that might be simple and easy to understand on their own, and you put them together, sometimes you get this kind of group action out of them that is very difficult to predict or impossible to predict from the individual component. Right? So think about flocking birds that do interesting things in the sky. You can't ever predict that, but just by watching one sparrow, it emerges from the group. And likewise in the brain, a lot of the sort of features that we sort of endeavor to study or understand can't be explained by the individual pieces and need to be considered as kind of a complex system, right? So let me just give you some evidence that the system actually impacts the disease. So here is a cartoon from a recent review paper that demonstrates this is a neuron, and this is also a neuron in blue. And the idea is that these are both diseased neurons, and they accumulate these red dots. And the red dots are meant to be that A beta molecule I mentioned. And these researchers found that if you drive activity, so you chronically stimulate this neuron on the right, you vastly accelerate and exacerbate the deposition of that A beta molecule. So you actually increase its production. So somehow activity in the system is sufficient to derail normal regulation of the brain and lead to disease. And I would sort of say a similar point that many people who work with mice know is that if you have clonally identical animals, so these are animals that genetically are the same exact animal, but two separate animals, and they're both destined to get Alzheimer's disease because you gave them some human tau transgene or something. They won't get it at the same time. One of them might get it at 4 months, and one of them might make it to 2 years before it can't move effectively, right? And in mouse years, that's a lot of time. And so there's clearly something about environment and activity that determines the rate and the severity and the onset of these diseases. We've had another hint that emerged in 2016. Hannah Icarino, a postdoc at MIT, showed that if you flash lights at a mouse 40 times a second, you can take, this is a diseased animal, A beta staining in its brain, and then if you flash these lights, you transition from this to this. So she showed that just by driving visual potentials, she could clear out A beta from the brain. And so there are some very serious caveats with that study, but I think that together these both point to a role for larger systems in controlling this. So if we go back to the car analogy for a moment, say your car's having trouble driving, you might be able to say, it's this widget or that part, but you might replace that and then run into the same problem over and over and over and over again. It's because you're driving with your e-brake on, right? And so you need to actually figure out what you're doing wrong in the larger system to prevent the kind of collapse in the first place. So with that as our analogy, I guess this is supposed to be a business community and we're supposed to raise the idea of challenges. So the big question is, well, then why is a systems view of neurodegeneration so hard, and why haven't we approached yet? So the big problem that I think that we face is having the technology and tools to even understand what the brain should be doing. We can understand what a neuron should look like, or maybe sort of the firing rate of those cells, but to understand what that kind of emergent property is that defines a healthy brain and thus allows us to identify a diseased brain is extremely challenging. So that's this question of what is the set point? If I walk into your house and it's 78 degrees, so what? If I know that your house is set to 68, I would suggest that you have a problem with your air conditioner. So we need to understand what the brain or these various circuits are set to before we can begin to really understand how to steer them back when they're diseased. Which then brings up the second point of how do we control that larger set point? How do you steer an emergent property within the brain to rescue it from kind of progressive pathology? So in an attempt to answer number one and address number two, my lab has developed a novel set of recording technologies that will let us get a view into that set point. So we are capable of recording from between 500 and 1,000 individual neurons spread across a mouse's brain. And we can do this in a freely behaving mouse for the longest recording that we've run at WashU is 15 months. So just contrast that, the typical approach to this type of recording might involve 16 to 64 neurons, and usually lasts from 30 minutes to maybe 24 hours. So these are categorically different things that we're doing, and it allows us to follow many, many, many neurons and brain regions across the entire course of disease, from health to onset into late stage disease, and begin to address these sort of dynamical set points that might define the disease. And so what this looks like on a mouse, here I was once told I can't actually show the mouse, so I made this drawing. This is a photograph that was then converted to a cartoon, but here we have 640 channels going into this mouse's brain. This is a mouse that had been implanted for many months and again, lived its happy little mouse life as we went to sort of record all of its behavior and all of its neural dynamics, and then ask when does it get sick and can we see that in the neural signatures. So skipping over a bunch of slides on theoretical physics, I want to show you what the set point is that we have identified. And we published this last year, and I don't know, I think it's cool. So there's an idea that systems poised at the, and it's air quotes here, edge of chaos are computationally ideal. And that line between stability and chaos is called criticality amongst theoretical physicists. So here's a visual to show you. So if we have a subcritical system, a system that's too stable, the parts within the system, so these little blue dots, they don't interact with their neighbors strongly enough to propagate a signal. So the signal just dies out. right? So I'm reseeding this, and you can see that the blue just kind of fizzles out. If you cross that line in the other direction and you make those interactions too strong, the little blue dots are too happy to interact with their neighbors, you end up with a supercritical system. And so in the supercritical system, we just see immediate saturation. And in both of these computational regimes, as you can imagine, there's really no information contained, because if you were to inject a blue dot, its chance of making it across that field is 0, right? On the left, it would just quickly fade to nothing, and on the right, it would be lost in noise. Now, in the middle, at exactly that phase transition between subcritical and supercritical, exists a regime in which everything is perfectly balanced, and you have ongoing dynamics at all spatial and temporal scales. You'll see short bursts and long bursts, 2 or 3 dots versus millions of dots, and they will never explode and never decay. And yet you have this sort of ongoing, sort of beautiful pattern emerges. And it's at this point that you actually, if you take it from a computer science theory, you maximize dynamic range, you maximize information transmission, information storage, memory, et cetera, et cetera, et cetera. They all fall out of this one computational regime. And we showed that this is evident in the brain. And when we push it, and we maintain that push for days, the brain will come right back to that point. So despite the fact that we've pushed and held that, it will find its way, it actively regulates itself and pulls itself back to that critical point. So we've identified this as kind of like the thermostat set point for cortical networks in the brain. Okay, so I know that I'm running out of time here. So let me show you how we would like to apply this and are starting to apply this to neurodegenerative disease. Here is our little mouse, and this is a raster plot. Hopefully you can see that it's sort of scrolling and animated, of many neurons. Each row is a neuron, and each dot is sort of its spike time that it's firing. We can analyze this data for those hallmark signatures of a critical system. So ignore this stuff because it doesn't matter right now. But the point is, we know that a healthy critical system should fall on this line. You can see the green text and the purple dots are healthy. And then what we can watch is that there's a deviation from criticality that progressively accumulates. Long before you can see the animal get sick, we start to see the neural dynamics separate from criticality. And that's this orange distribution. And so then we measure that deviation. We call it DCC, the deviation from criticality coefficient. But basically, we watch that error accumulate, and it predicts that the animal is going to get sick. So then what we'd like to do is take these regimes and pass them into a model to try to figure out what could possibly give rise to those. And so we go through 20 million models. And the models produce maybe out of 20 million, 12 or 15 proper solutions. So then we can ask, well, what's the defining feature that keeps all of those solutions together? And it might be, I don't know, in this case, something special about the FS basket cell, right? So we can identify that. And then we can actually take genetic control over those FS basket cells and push on them back in the original animal to try to restore dynamics, because we've identified these as causal in the shift from purple to orange. So that's all great and may work in a laboratory, we hope. But the challenges and hurdles, which I've been asked to raise, basically, we need to scale this up in a mouse. So we need better technology that's easier to build, that can sort of give us these recordings. And so we're not the only group in the world doing this. And then obviously we need to test these core theories in humans. It's certainly not hard. I know how we can do it. It doesn't have to be invasive, but it takes resources and people and sort of collaborative groups. And then obviously intervention is sort of the final step. So if we can recover or reverse a disease in a mouse, then you would move towards doing that in a human subject. And much of this is dependent on the methods and the technology. And that is where Shantanu will take over. But with that, I should quickly acknowledge some folks in my lab. So this is my group here. And these are all fantastic people. WashU has exceeded all of my expectations, largely because of who we have here and the sort of quality of the community. And then also I should very, very much point out Ralph Wessel is a theoretical physicist, and Zheng Ma was his graduate student. And these two are the ones who introduced me to the ideas of criticality, and we took it and ran with it. And so we published a very nice paper with them last year, again, demonstrating that criticality is a set point in the healthy brain. And obviously I should thank the people who give me the money to do the things that I do. So with that, I guess that's 17 minutes and I'm out of time. This is where Zoom is awkward because it's just silent.
So again, anyone who wants to ask a question, please unmute yourself and
maybe I'll ask a question if that's okay.
Take it away.
Thanks, Keith. That was fun. I like your way of presenting that and your theory there. I was wondering, given that you've presented this criticality point as kind of an emergent property of the brain as a whole, how do you know whether it's the right thing to look at, the right emergent property to look at, and do you think it might differ for different disorders? Do you think like this might be really relevant for Alzheimer's but maybe less so for psychosis or something like that?
Mm-hmm. I guess I'll just go out on a limb here. Like, this is clearly speculative, but given that so many of the kind of desirable computational features of a network fall out of that critical regime, I would suggest that I think that criticality is the thing to look at because it predicts, for example, gain control, or it predicts sort of branching ratio. All of those things have to work for the system to be critical. So whether or not the brain is actually measuring criticality or some other feature that inevitably produces it doesn't really matter at the end of the day if those things are always mapped one-to-one, because then controlling criticality will give you control over whatever those underlying aspects are. But so then your second question, could there be disorders in which criticality is sort of irrelevant? I mean, sure. So you could imagine a situation in which a system is critical, but it's just what the attractors that it's drawn to for various processing features are just pathological, right? So say psychoses, where you don't have any problem in the ability to sort of transmit and maintain information flow, you just have really messed up information. So I can certainly see that being the case, but we know that in many brain regions, we see signatures of criticality. So that's one of the advantages of being able to record all over the brain. And also in a couple different disease models, we've seen issues with criticality. So in 3 different autism models and in a tauopathy model, so not AD, but just sort of broad neurodegeneration, and then also one of these sort of APP overexpression models that's specific to AD. They all have a similar dynamical signature at the level of criticality. And so then the question is, are the underlying pieces that are starting to kind of fray, are those shared, right? Because as I mentioned, like, you got to consider this more complex system. And so there could be 5 or 6 or 10 or 100 different components that all contribute to the larger sort of dynamical phenotype. And so figuring out which ones to push on becomes the important question.
Nice, thanks.
Thank you.
Excuse me, I had a brief question on something I was a little unclear about for the DCC.
Yeah.
So regarding how it's calculated, is it directional for the subcritical or supercritical, or is it based on whether it's critical or non-critical?
Right now, it's a scalar value, so it's just absolute value. It does not tell you sub or super, but that's very easy to figure out. So without getting into too much detail, DCC emerges from from step 3, and step 1 and 2 that you have to take very much indicate sub or super criticality. So if you see that DCC is off, you can just check back to the input steps. But DCC itself is like it just tells you, it's like distance, so it doesn't tell you the direction at all.
Okay, thank you.
Yeah. And also, I will just point out the only time we've seen a supercritical regime in an intact brain is during like a grand mal seizure, right? Because that kind of runaway expansion of activity is it's nearly lethal. It's definitely bizarre. But we see a lot of subcritical signatures in pathologies or sleep deprivation or other things like this.
I was definitely wondering that. Thank you.
Yeah. Shantanu, do you want to do your thing?
I don't have any questions, but yeah, sure. Meaning I can.
Well, let me ask one more question before we go. You you said the long-term idea is to find ways to push people back into criticality. But
Yeah.
I wondered if before you get to that, whether this might be a early diagnostic tool for AD or something.
Yeah, Rob, that's a very, very good point. So I think that preliminarily, with the animal models that we're working with, using molecular markers, you could diagnose the animal at, say, 6 months. Using the critical dynamics, we've been able to diagnose animals as early as 2 months. So that's the equivalent of taking a human patient at 25 or 30 years old and saying, hey, no neurons have died, you've lost no synapses, but you're gonna have Alzheimer's in 15 years or 20 years. So we need to start doing something now. So at the bare minimum, if this were to hold, even if we don't get a new mechanism to push on, we would have potentially the most sensitive sort of diagnostic biomarker that we could apply very early in life to then do whatever else it is that needs to be done. My hope, though, really is that it's not purely a linear downstream result of the thing, but that manipulating these systems will feed back on the underlying processes. And I think that the data that I showed you, or the cartoon data, where people drive activity in neurons and that exacerbates the production of A beta. When you drive a bunch of neurons exogenously like that, and you put in optogenetics, you flash a bunch of lights, you push the system right out of a critical regime, and you increase firing rates. And so the question is, can we identify how we need to manipulate them and which cell types we need to manipulate? But I guess I just have a strong hunch that it won't purely be diagnostic. I hope. I don't know. I'm probably wrong. I frequently am wrong.
Hey guys, could I ask a quick question?
Yeah.
And I apologize. I jumped in during your presentation, so if I ask a dumb question here, sorry.
Okay.
I'm trying to understand in terms of, like, are you trying to get, say, a baseline, like a fingerprint for what a healthy brain would look like in terms of its neural activity, and then basically, yeah, defining when a healthy brain starts to veer off to one side or another, and that would be sort of the definition of it's going into, you know, an Alzheimer's state? Does that make sense?
So that's a good question. So we published a paper last year in totally healthy animals, sort of demonstrating that this critical regime is stably expressed, and that when we push on it, the system comes back. So it'd be like if you opened up your doors and left them open in February, that you have a furnace to kind of restore the system to that set point. So we sort of demonstrated that criticality seems to function as a set point for the kind of emergent dynamics in the cortex. And then, as you point out, yeah, the idea would be that if you could detect deviations and sort of hops in this critical regime early, they sort of go above what you would expect under sort of normal noise or sort of random variation, that maybe then you could have some insight to say, okay, this is starting to sort of break away from a normal trajectory and indicate an impending problem. And then hopefully, as I said to Rob, actually be able to manipulate that thing and maybe affect the course of the disease.
Are you familiar? I'm familiar with a paper published by, I think it was the Imperial College London, on the entropic brain. Are you at all familiar with that paper or theory?
Probably, yeah. So that's entropy is maximized at criticality. So these things are all very, very related.
Okay, so hey, points for me for thinking,
yeah.
thinking they're related. On that paper you published last year, is there a way for me to read it?
Yeah, I mean, technically it's illegal, but not illegal, but it's sort of
not illegal now.
It's against journal policy, but we just put PDF on. If you go to our lab website, you can click it and download it.
Fantastic. Don't even have to send an email. Awesome.
Nope.
Appreciate it.
So Keith, can I ask you a question?
Go for it.
So do you view then learning as a change of the set point? Like if you keep applying a stimuli and you keep changing the criticality, does it mean changing the set point is equivalent to learning?
I don't think so. So, and we have some preliminary data on this in the lab. So if we take, so for those of you who are not neuroscientists, there's something called optogenetics, where you can express a light activatable channel in a neuron so that shining light on it drives the neuron. So we express that broadly in cortex, and we're recording, and we blast a region for 3 or 4 or 5 hours, and you actually start to change the connection strength between neurons. And we believe this is the biological sort of mechanism of learning. And when we do that, we see that as we're blasting this, we push the system away from criticality. And then once it recovers, interesting, the animal needs sleep for this, but once it recovers to criticality, so that the set point is the same then, but the pairwise correlation matrix, right? So basically in eigenspace, the network's position has changed dramatically, right? So the way that the neurons are interacting has changed, the pairing of neurons has changed, but despite that sort of imposition of a new architecture, they still restore themselves underneath that kind of critical umbrella. I think that, well, we can get into that later. I've got a lot of thoughts on this.
So this